Good morning everyone, my name is Krishna Chaitanya and I will present our IBS2 project which is graph-based detection of mutations in DNA sequences which I have worked with my teammates Akhil and Vinay. This project combines bioinformatics and graph theory to improve how we detect mutations in DNA. In this project, our goal is to develop a method that uses graph structures to detect and visualize mutations in DNA sequences. Traditional tools often rely on a single reference genome, which introduces reference bias and may overlook complex mutations. By using graphs, we can capture multiple sequence paths and also highlight differences more clearly and make the analysis scalable, and also we can use large data sets. We also implemented a demo that detects mutations and also visualizes them using Python. Let's begin with the basic understanding of DNA mutations. DNA is the blueprint of life, made up of 4 nucleotide bases A, T, C, and G. These bases are arranged in specific sequences that carry genetic information. A mutation is any change in this sequence. It can be as small as a single base substitution, which is also known as single nucleotide polymorphism (SNP), or as large as a segment being inserted, deleted, or inverted. These changes can happen due to errors during replication, exposure to radiation, or environmental factors. Mutations are significant because they are leading cause of genetic diseases and they also drive evolutionary changes across the generations. Therefore, detecting them accurately is extremely important in medical and genetic research. Traditional looks like BLAST or BWA align test sequences to a linear reference genome. This method assumes there is only one correct path or sequence, which introduces reference bias, and this can also cause the tools to miss complex or rare variations. Graph-based models offer a better solution. Instead of a single path, they can represent multiple and also alternative genome paths. This allows them to handle structural variants and population level diversity much more effectively. Additionally, graph-based methods are both scalable and parallelizable, which makes them ideal for analyzing large scale, and also they put high throughout the sequencing data. This is why they are becoming increasingly popular in modern bioinformatics. Our objectives are very clear. First, we aim to detect mutations using graph structures. Then we visualize DNA sequences as directed graphs to see these mutations clearly. We also calculate similarity scores between sequences and compare the effectiveness of graph-based methods versus the traditional ones. Our first step is to take a reference DNA sequence and also a test sequence. For wherever there is a difference found, we flag that position as a mutations. We can take various examples and then we compare them base by base. Next, we convert the sequences into graphs. Each base is a node labeled with its index and letter like 0 underscore A, 1 underscore T, and so on. Edges connect each base to the next. Then we construct separate graphs for the reference and test sequences. Mutated nodes in the test graph are marked with a flag, so we can easily identify them later. Once we know the mutations, we represent each DNA sequences as a directed graph. We bring this graph to life using visualization tools. Blue means original, green means unchanged, and then red highlights the mutations. To give a numeric data of how different the sequences are, we also calculate how many matching short patterns they share. We also score how similar the two strings are comparing by short pairs like AT or GC. Finally, we calculate how similar the two charts are based on how many letter pairs match. At this stage, we have developed and tested a Python script that can detect mutations by comparing a reference and the test DNA sequence. We also built the visualization model using NetworkX for graph construction and Matplot for plotting. The program correctly identifies mutation points and marks them visually. So far we have made a great process. It's simple, clean, and works well with small DNA strings. Our visual output shows the reference sequences in blue, test sequence in green, and any mutations in red. This clear visual representation makes it easy to pinpoint difference between the sequences. It also outputs a similarity percentage which quantifies how closely related the sequences are. Blue means the original DNA, green means there is no mutation, and red is where the mutation occurred. It even gives a similarity score to show how much the two sequences have in common. There are still some areas we plan to improve. First, we want to extend the tool to handle longer DNA sequences. We also aim to create more efficient graph layouts for large datasets and improve performance and scalability for the real world. DNA strands and also improve the design so it stays readable even without a lot of data. Uh, we aim to scale the system for longer sequences, improve graph rendering performance, and also to explore parallel processing techniques to speed up the computation on larger datasets. To summarize, our graph-based approach to mutation detection offers it clearly visually representation and accurate identification of differences between the DNA sequences. We have shown that this method works well in practice and also provides both clarity and scalability for genomic analysis. We have also successfully built a working Python script that detects mutations between DNA sequences. The visualization module developed using NetworkX and Matplot clearly shows mutation locations and also allows us to compare sequences visually and numerically. The system works reliably for short DNA inputs and helps to illustrate sequence similarity in user-friendly way. Future work includes addressing scalability, handling longer sequences without overwhelming the graph, which requires layout optimization. We also aim to parallelize portions of the script to enhance performance when processing real genome data and speed the improvements using optimized data structures and multiprocessing wherever applicable. We refer to core sources like genome sequencing literature, NetworkX documentation for graph generation, and we also have done research on debrugging graphs in genome assembly. Thank you.